Hello everyone and welcome to the new session of the lecture series. And in today's lecture, we'll be revising the chapter Plant Growth and Development. So today we'll be studying about the plant hormones, also known as phytohormones. Alright, so we'll study about the discovery and the function of the hormones, and then we'll study about some of the terms such as vernalisation and photoperiodism. And the rest of the topics we have already discussed in the previous lecture. Okay, so we'll discuss everything in brief here. All right. So let's start with the plant growth regulators. Okay, now you already know that plant growth regulators or plant hormones, they are of 5 types. Okay, first are the indole compounds. Okay, indole compounds have indole group as a parent nucleus. Example is indole 3-acetic acid. Okay, then we have adenine derivative. What is adenine? Adenine is a nucleotide. Or adenine is a nitrogenous base. Okay. You've seen adenine in DNA. Okay. We have adenine, guanine, cytosine, thymine. So example of adenine derivative hormone is kinetin. Okay. Kinetin has this N6-phosphorylaminospurin. This is known as kinetin. Now, next are the carotenoids derivative. Example is abscisic acid, also known as ABA. Then we have terpenes derivative, which is gibberellic acid. Okay, first gibberellic acid, okay, isolated was GA3. And then we have the gaseous hormone, which is known as ethylene and has the formula C2H4. Ethylene is also known as volatile hormone. Now, the plant growth regulators are divided into 2 groups. Okay, first group is the plant growth promoter. Okay, so within this plant growth promoter, what are the examples? Auxin, gibberellins, and cytokinins. These are plant growth promoters. Alright, and what are the functions? These hormones are involved in the cell division, cell enlargement, or you can say cell elongation, pattern formation, trophic growth. Okay. You can say like phototropism, gravitotrophism. Okay. And then they are also involved in flowering. Okay. Flowering, fruiting, and seed formation. Now the group 2. Within group 2 are included the plant growth inhibitor. Okay. Example of a plant growth inhibitor is abscisic acid. So abscisic acid is a plant growth inhibitor. So, okay, and what are the role of this or where they are used? These type of hormones, plant responses to wounds and biotic and abiotic stress. Okay, so they are involved in plant responses to wounds and biotic and abiotic stress. So biotic stress is caused by the suppose microorganism and pathogens. And what are the abiotic stress? Caused by the like environmental stress such as drought. Okay, or you can say salinity or high temperature. So these are abiotic stress. So these hormones, okay, are also involved in various growth inhibiting activities such as dormancy and abscission. Okay, you know these terms dormancy and abscission now. Dormancy is the temporary, okay, inhibition of the physical activities of the seed, you can say seed or the bud. Okay, there can be seed dormancy or bud dormancy. Alright, so what happens? There is a temporary inactivation of the physical activity of suppose say seed. Of the seed. Okay. And what happens if there are favorable conditions are given, then the growth will resume. And what is abscission? Abscission is the simply you can say falling off of the organs of the plant, or you can say detachment of the suppose the organs of the plant such as leaves, or you can say fruits or flower. Okay. So this was about the group 2 hormone. Now there is one more hormone, ethylene, which is the gaseous hormone. This can act as a growth promoter and growth inhibitor also. Now let's talk about the discovery of the phytohormone, and let's start with the hormone auxin. So Charles Darwin and his son Francis Darwin. So what they observed that the coleoptile. Okay, what is coleoptile? Coleoptile is a sheath covering the shoot tip. Alright, so coleoptiles of the canary grass, okay, they responded to the unilateral illumination by growing towards the light source. Okay, so this is the coleoptile, okay, or you can say this is the shoot tip of canary grass. They were working on canary grass. Okay, if it was illuminated with light, so what happened? There was a bending or there was a unilateral movement, okay, towards the light source. Alright. Okay, and if this tip was removed, okay, so there was no movement. Okay, like this only straight, even after illumination by light. Alright. So now, with this experiment, they concluded that there is a presence of a substance that guides canary grass grow towards light. So they said that there is something in the shoot tips, or you can say coleoptile of the plant. Okay, there is some substance present there. Okay, which helps in the movement of the plant. Okay, towards the light. And later, FW went. What he did? He isolated this substance and named it as auxin. Okay, from the tips of the coleoptile of oat seedling. So he was working on oat seedling, which is also known as an Avena sativa. Okay, oat is known as Avena. Alright, so what he did, if this is the shoot tip, okay, of the coleoptile or the coleoptile of the oat seedling, okay, this is oat. So what he did, he cut this shoot tip, okay, this region, and he placed it on the agar. Okay, this is agar. Okay, and later what happened? After some time, substance from this, okay, there was some substance here in the shoot tip. Alright, this substance was present here. I'm denoting it with this red. Okay. So what happened? This substance then diffuses into the agar. And the agar block became red. Okay, here this substance diffuses into agar. Alright, then what he did? He placed this agar block on this coleoptile, of which shoot tip was excised. He placed this agar here. Okay, this contains this red substance, and this, okay, transferred here. This diffused into the, okay, in the shoot tip or the axis of the plant. Then what happens? If it was illuminated by light, so there was a movement again here. Okay, block was kept here. The substance has diffused here, okay, in the tip region, and there was a movement seen. So this is how auxin was discovered by FW Wendt. This is known as agar block experiment. Agar block experiment. Now, how gibberellins were discovered? So, Bacne disease, okay, also known as the foolish seedling disease. Okay, this disease was seen in the rice seedlings. And what was the cause of the disease? A fungal pathogen, Gibberella fugicori. So, this fungal pathogen causes a disease in rice seedling known as Bacne disease, and is also known as the foolish seedling disease. Why it is called foolish seedling disease? Because there is an enhanced growth or the increased growth of the internodal region of the stem. Okay, or you can say of the main axis. Internodal region of stem. Okay, so now they observe that why there is an increase in this internodal region. It was because of the presence of a substance which was released by the fungus. Okay, it was released by the fungus Gibberella fugicori. Okay, and this substance was later identified as gibberellins. Alright. Okay, this is clear. So, what happened? E. Kurosawa, he was a Japanese scientist. What he did? He did an experiment in which he treated the uninfected rice seedlings with the sterile filtrate of the fungus fugicori. Alright, sterile filtrate means they contain only the spores of the fungus or whatever, extract of the fungus. Alright, and nothing else, no other microorganism. So he treated the infected rice seedling with the sterile filtrate of the fungus. And what he reported? He reported the appearance of symptoms of this disease. Means these seedlings were also infected. Okay, and later the active, and this also, okay, disease was also appeared in the uninfected rice seedlings. Means there was an increased growth of the internodal region. Okay, and the active substance was later identified as the gibberellic acid. The next is cytokinin. So Skoog and colleagues, what they were working on the tobacco internodal segment. Okay, so what they observed that the callus growth occurred in the tobacco internodal segments only when auxins. Okay, first auxin was supplied. Okay, only when auxins were supplemented with specific nutrients obtained from extract of either vascular tissues, yeast extract, coconut milk, or the DNA. Okay, so they said that the callus initiated in the tobacco stem internodes only when, along with auxin. So auxin is important, but along with auxins, specific nutrients. Okay, they were also supplied. Okay, and these specific nutrients were obtained from vascular tissue, yeast extract, coconut milk, or the DNA. Then only there was callus formation. Okay, and then the substance from these specific nutrients were isolated. Okay, so these additional substances, first of all, they triggered the proliferation of the callus. Okay, means they initiated cell division. Okay, and this substance was identified by Skoog and Miller. So Skoog and Miller identified and crystallized a substance that promotes cytokinesis, means cell division, and which they named as kinetin, which is a cytokinin. Now let's talk about abscisic acid. Okay, so abscisic acid was discovered actually 3 times by 3 independent researchers. Okay, and what they did, they named this, okay, these substances as, first one named it as inhibitor B. Okay, and then it was named as abscission 2, and it was also named as dormin. Okay, so abscisic acid was discovered 3 times by 3 individual researchers. Alright, and later what happened? All these inhibitors, okay, so they showed inhibiting activity, growth inhibiting activity. So all these inhibitors were chemically identical. Okay, when they characterized them, so they was chemically identical and they were named as abscisic acid, ABA. Now next hormone is the ethylene. Okay, so cousins, okay, they confirmed the release of the volatile substance from the ripe oranges. Okay, so they said there was a substance, volatile substance released from the ripe oranges that accelerate the ripening process of the stored unripe bananas. Okay, means this substance was able to ripe the bananas. Okay, helped in the ripening process of bananas. So later this volatile substance was, okay, identified as ethylene, a gaseous plant growth regulator. Okay. Now let's talk about the functions now. Okay. Of the phytohormones. We'll do it, okay, briefly. So auxins, okay, is derived from the word auxine, which means to grow. It was first extracted from the human urine. It is primarily synthesized in the growing tips of stems and roots, means shoot apical and root apical meristem. There are two types of auxins, natural and synthetic auxin. Example of natural auxin is indole acetic acid, indole butyric acid. Synthetic auxins are naphthalinic acetic acid and 2,4-D, 2,4-dichlorophenoxyacetic acid. What are the functions of auxins? First important function is the development of root, and it also promotes flowering. Next function is the premature shedding of the young fruits and leaves. But what auxin do? It promote the abscission. Okay, means detachment of the mature leaves and older fruits. Okay, auxin also help in cell division and cell elongation, and it promotes apical dominance. This is very important. What is apical dominance? When the apical bud suppresses the lateral bud, or you can say the lateral growth. This is known as apical dominance. Okay, and a phenomena known as, there is a term known as decapitation. So decapitation, what does decapitation means? There is, we remove the apical bud. We remove the apical bud which contains the auxin, and this promotes the lateral growth. So this is seen during tree plantation and hedge formation. Promotes the lateral growth. Okay, another important function is parthenocarpy, which helps in the generation of seedless fruits. Okay, this is seen in tomatoes. Parthenocarpy is formation of fruit without fertilization. Okay, now auxins also have herbicidal activity. Okay, so auxin, what they do? They kill the dicotyledonous weed, but they don't affect the monocotyledonous plant. Okay, so that's why they are used to prepare the weed-free lawns by the gardeners. An example is 2,4-D, 2,4-dichlorophenoxy acid. So auxins also have commercial importance. Okay, they have commercial value. Alright, and auxins also control xylem differentiation. Okay, so xylem can be differentiated into vessels and tracheids and xylem parenchyma and xylem phloem. Okay. Now let's talk about gibberellins. So there are more than 100 gibberellins. Okay, they have been named as GA1, GA2 and GA3. The first gibberellin to be discovered was GA3, and most of the gibberellic acid are acidic. Now, functions of gibberellic acid, first is increase in the length of the axis. Alright, it also speed up the malting process in the brewing industry. Okay, this is important. And what does gibberellin do? Cause fruits like apple to elongate and improve its shape. Another important function of gibberellins are they hasten the maturity period in conifers. Means they help to attain the reproductive stage earlier. Okay. And what does it lead? This leads to the early seed production. One more thing you have to remember is that gibberellin promotes bolting. Bolting means internode elongation just prior to flowering. And these are the examples. Now let's talk about the function of cytokinin. Now kinetin, which is a synthetic hormone. This is synthetic. Okay, this was first discovered from herring sperm DNA. This you have to remember. And then later zeatin, which is a natural hormone. So zeatin was discovered from the corn kernels and the coconut milk. Now where are cytokinins synthesized? They are synthesized in the growing regions. Okay, or you can say growing regions of the root. Okay, such as the root apices, root apex, developing shoot buds and young fruits. What are the functions of cytokinin? First is the lateral shoot growth. Okay, they help or they promote. They promote the lateral shoot growth and adventitious shoot formation. What are adventitious shoot? Okay, shoots which form from the internodal region. Okay, usually the stem arises from the nodes. Okay, but if the stem arises from the shoot arises from the internodal region, then it becomes an adventitious shoot. Cytokinin also help to overcome the apical dominance, means it promotes the lateral growth. Alright. And one of the most important function is it delay the senescence. Okay, senescence. What is senescence? It is the known as biological aging. Okay, senescence ultimately leads to death of the organism. Okay, so cytokinin delays senescence. How? By promoting the nutrient mobilization. So what happens? Suppose this is a seed, this is a leaf, and if we apply cytokinin here in this region, in this region we have applied cytokinin. So what happens? Cytokinin will divert all the nutrients to this region. Alright, and this region will remain green and healthy, whereas the other regions will become yellow. Okay, so cytokinins, they promote the nutrient mobilization. Okay, and this is how it delay the senescence. So during senescence, what happens? There is oxidative damage. Okay, there is chlorophyll degradation. Okay, due to chlorophyll degradation, what happens? The leaves become yellow. Okay, photosynthetic apparatus deteriorate. Photosynthetic apparatus also deteriorate or disintegrate. Okay, degenerate. So all these things happen during the senescence, and cytokinin, what it do? It will reverse all these effects. Alright. Another role of cytokinin, help to produce new leaves and chloroplast in the leaves. Now let's talk about the ethylene. So ethylene is also known as the ripening hormone. Why? Because it helps in the ripening of the fruits. Okay, so it is a simple gaseous hormone produced by tissue undergoing senescence and ripening fruits. And this you have to remember. Ethylene, what it does? It increases the respiration rate during the ripening of the fruits. Okay, and this rise in the rate of the respiration is known as respiratory climactic. Okay, so rise in the rate of respiration during ripening, okay, which is caused by ethylene, is known as respiratory climactic. Okay, functions of ethylene, again, promotes horizontal growth of the seedling. Okay, so if this is a seed, okay, plant is growing like this. If stones come in between, then it promotes horizontal growth. Okay, if this is a stone till here, plant will grow like this horizontally, and again it will go upwards. Okay, so promotes the horizontal growth. So this is the stone. Promotes the horizontal growth. It also promotes the swelling of the axis, and it promotes the apical hook formation. Okay, in dicot seedling. Okay, apical hook is formed due to the differential cell division, which is caused by the ethylene. And what is the role of apical hook? It protects the shoot tip. Alright. Another important function is it promotes senescence, means growth inhibitor. It promotes senescence and abscission of the plant organ. And what does else ethylene does? Ethylene breaks seed and bud dormancy, means helps in the growth, means it is a growth promoter now. Okay, so ethylene was a both growth inhibitor and a growth promoter. So you can see now how it is a growth promoter and a growth inhibitor. Some of the other functions are helps the leaves and the upper part of the shoot to remain above water. Okay, how does it promotes? How does it help the leaves and the upper part of the shoot to remain above water? By promoting the internode elongation. For example, in rice. Okay, Another important function is promote root growth and root hair formation. Okay. So if there is more root growth and root formation, it will increase the surface area for the absorption. Surface area for the absorption of what? Absorption of water, minerals and nutrient. You can write a increase. Alright. Another important function is used to initiate flowering and for synchronizing fruit set, okay, in pineapples. Now you have to remember this ethphon. Ethphon is a commercial product. Okay, it is widely used as a source of ethylene. Ethphon, what does it? Hastens the fruit ripening. Okay, same as ethylene in tomatoes and apples, and accelerates abscission. Okay, promotes abscission in flowers and fruit. Okay, and also you can say thinning of the cotton, cherry and walnut. Okay, what is thinning? Thinning is the removal of the, okay, some of the leaves or the parts of the or the organs of the plant so as to make space for the other organs. This is known as thinning of. Okay. Now again, ethphon, what does it? Also promotes the female flowers in cucumbers. Okay, thereby increasing the yield. Okay, if there is an increase in the female flower, okay, the ovary will modify into fruits and all, seeds will be produced and yield will increase. Now let's talk about the phototropism. Now what is phototropism? Phototropism is a physical response of the plant to the relative length of day and night. First you have to remember. Okay, relative length of day and night, or you can also say duration of light and darkness. Okay, we know a day, day consists of 24 hours. Okay, now we can divide it into 12 and 12. Okay, 12 hours light and 12 hours dark. Okay, or we can also divide it like this: 14 hours light and 10 hours dark. Okay, so these are the duration of the light and the darkness. Okay, or we can also say relative length of day and night. Alright, the photoperiodism is nothing but the physiological response of the plant to the relative length of day and night, okay, which influence various development and reproductive processes in plants, such as flowering and dormancy. Okay, now there was a hypothesis given that a floral hormone, okay, or you can say a substance is present in the leaves. Okay, so leaves, okay, so light is perceived by the leaves. Okay, sunlight is perceived by the leaves with the help of what? There is a photoreceptors known as phytochrome. Alright, so hypothesis was given that there is some substance present in the leaves. Okay, suppose this substance is produced in the leaves. This substance travels to where? Shoot apical meristem. And what it does? It causes flowering. Okay, a substance from the leaf. Okay, so this substance was named as florigen. So florigen is a floral hormone. Alright, and it is a proteinaceous hormone. Okay, made up of protein. And this hormone is involved in flowering. It initiates flowering. That's why it has been given the name florigen. And it is produced in the leaves and transferred to the meristem, shoot apical meristem. Now let's talk about the long day, short day and day neutral plants. Alright, so what are long day plants? Plants that begins flowering when the duration of the daylight exceeds the critical threshold, or you can say the critical value. Alright, suppose again a day is of 24 hours. Okay, if the critical if the critical threshold is 12 hours. Okay, this is the critical threshold. If the long day plants are given more light, okay, or they are kept in the day for more than this 12 hours, suppose for 14 hours. Okay, then only they will flower. Okay, But if the photoperiod is less than 12, suppose they are given 12 hours of daylight, then it won't flower. Alright. So again, coming back to long day plants, plants that flower when duration of the daylight exceeds the critical threshold. So in this figure, you can see critical photoperiod. You can also say critical threshold, critical photoperiod, or the critical value. Alright. So if the day length is more than the critical photoperiod, then only it will flower. Otherwise, it will not flower. Examples of long day plants are pea, sugar beet, cabbage, wheat, and radish and spinach. Now let's talk about the short day plants. Okay, so it is opposite of long day plants. Plant that initiate flowering when the duration of the daylight is shorter than the critical photoperiod. Okay, again, if the critical photoperiod is 10 hours. Okay, so duration of the daylight should be less than this critical photoperiod. Okay, so if 8 hours of daylight is given, then only it will flower. Okay, alright. Examples are chrysanthemum, rice, soya bean, onion, cotton, and soya bean. Again, so this is the short day plant. This is the critical photoperiod. They will flower, okay, only when the duration of the daylight is less than the critical photoperiod. Only in this condition it will flower. Now third one is the day neutral plants. Okay, so day neutral plants, they are independent of the length of the light exposure. Alright, and they will only flower, okay, after the vegetative maturity. Okay, flowering after vegetative maturity. When they will reach a certain stage, only then they will flower. Means they will transform into reproductive phase. Reproductive phase, what happens? Flowering takes place. Alright, examples are tomato, cucumber, cotton, dandelion, and sunflower. Now next term is vernalisation. So during vernalisation, what is the process? The process in which the plants are exposed to prolonged period of low temperature. Okay, and due to this exposure of the low temperature, there is initiation of flowering. Okay, so it is a method of flowering by giving a short period of low temperature. And what does vernalisation do? It prevents the premature reproductive growth. Alright. So if there is premature reproductive growth, plants won't be healthy. So it prevents the premature reproductive growth. Examples of vernalisation, we can see some of the fruit crops like wheat, rye, and barley, they are cultivated twice. Okay, biannually. So spring varieties, they are sown in the spring and reaped at the season's close, whereas the winter varieties are planted in autumn, okay, and harvested in midsummer. Now winter varieties, they require low temperature. Alright, so if the winter varieties are sown during the spring, they won't grow and they will not flower. Alright, so what happens? A low temperature treatment is given, okay, if they have to flower in the spring season. Alright, so if the winter varieties, okay, they are grown in the spring, they will not flower. Okay, so what happens? A low temperature treatment is given. Okay, and then they can flower. Alright, another example is some of the biennial plants such as cabbage, sugar beet, and carrots. They also require a short period of cold temperature to initiate flowering. Okay, so this was about the vernalisation. So this was the end of this chapter. We have revised all the topic briefly, and thank you for listening. We'll meet again in the next session.